So next up we have Isobel Guthrie from the University of Oxford. Thanks, Isobel. She's going to speak about the dynamics of indels at a within-host level during persistent SARS-CoV-2 infection.
Okay, so hi everyone, I'm Isobel Guthrie and I'm a DPhil student in the Pandemic Sciences Institute at the University of Oxford. I'm supervised by Katrina Lithgow and Matthew Hall, and my DPhil is all about indels in the genome of SARS-CoV-2 and their role in its evolution. So the project I want to talk to you about today is looking at deletions in persistent infections. So why should we be interested in deletions in persistent infections? Over the past 5 years, we have seen repeated emergence of new divergent variants of SARS-CoV-2. So Alpha in 2020, the emergence of the Omicron variants in 2021. And when we're examining the dynamics of the virus at this kind of global level, we also see some really interesting patterns when it comes to deletions. So this is the 6970 deletion, which is in the N-terminal domain of the spike protein. And what we see with this is this interesting switching dynamic. So each major wave with 6970 is followed by a wave without 6970. So in addition to this, and a bit less well noted, is the 144 deletion, also in the N-terminal domain of spike, and this shows a similar switching dynamic to 6970. So our leading hypothesis for where these new major variants of SARS-CoV-2 emerge from is that they emerge during persistent infection. So using a data set of persistent infections collected through the ONS COVID infection survey, I want to ask, do we observe de novo deletions emerging during persistent infection? And are they the same as these lineage-defining deletions that we observe at a global scale? So essentially, are these deletions that we see emerging during persistent infection? So my work is building on Mahan Ghaffari's work. He presented here last year, I think, about his detection of persistent infections in the ONS COVID infection survey. So I'll just summarize quickly. Mahan detected 576 persistent infections in the ONS COVID infection survey. These were defined as infections with at least 2 sequences 30 days apart. The majority of these lasted between 30 and 60 days, but as you can see, some of them lasted a very long time. For example, this one lasted over 300 days. So Mahan has also looked at the dynamics of single nucleotide variants during the course of infection. So on the right here is an example of a persistent infection which lasted over 150 days, was sampled 4 times. And so on the x-axis, we have within host allele frequency. So you can see 0 to 1. So this represents how many reads do we see that have the given single nucleotide variant. Y-axis is distance per sequence. So what we observe at a within host level during persistent infections when it comes to single nucleotide variants is that we see some emerge at a sub-consensus level before disappearing, others will emerge and fix. So what about deletions in this context? So the thing with deletions is that they're actually quite hard to deal with. They're difficult to consistently define. So we started with a position-by-position analysis. And what I mean by this is we go through the sequence position by position, and we see how many reads aligned to this position have a gap character there. But this is really not an ideal approach because of the nature of alignments. So sometimes there's just not enough information at the edges of reads for the reads to be aligned correctly. So for example, these sequences here with the stars, they align well as is, but they can also align quite well shifted along with gap characters in the middle. And the thing about this is that there just is not enough information to be able to say. So this can lead to a false signal of within host diversity at these positions. So our approach is to use phyloScanner, and to do this, we define windows around a suspected deletion of plus or minus 10 base pairs. phyloScanner then extracts only the reads which span this entire window. And from this, you can get a much cleaner read of what the deletions look like, and you can also detect deletions of different lengths which span in the same region. So results. So what I'm going to present here today is my de novo in-frame deletions, which reach above 20% in any given sample. And de novo is defined as being at below 5% frequency in the first sample. So across all the persistent infections, where do we see deletions emerging? So hopefully you can see there are two quite obvious hotspots. In non-structural protein 1. And this is quite interesting because the deletions that we see in this aren't actually very common at the within host level. So we see 19 deletions of variable length between codon positions 80 and 85. The most common of these was del 81 to 85. So the effect of these deletions is actually a bit unclear. A longer deletion spanning the region has been associated with a reduction in interferon response, but the actual effect of these deletions, why it's occurring during persistent infection, isn't something that we really understand yet, and some further work is needed on this. So our next hotspot is in spike, but specifically in the N-terminal domain of spike. So the two deletions that I mentioned earlier, del 144 and 6970, actually we observed both of these emerging de novo during persistent infection. So del 144 is actually the most common deletion that emerges in the persistent infections, emerging 10 times. You can see that on the right here, these purple lines all represent the trajectory of del 144 during a persistent infection. We see the emergence of 6970 in a single persistent infection. This one actually stays at a subconsensus level during the time period sampled, so it only reaches around 30% frequency within host. We also see emergence of this del 243, 244 in 4 persistent infections. And what's interesting about this one is that it's actually lineage defining for the beta variant, which was a variant of concern back in 2020. So when we taking all of the deletions in the N-terminal domain and then mapping them to structure, it becomes a bit more clear what's happening. So the N-terminal domain has these flexible loop regions called N1 to N5. And actually, when we map our deletions to the structure, what we see is that these deletions are emerging inside these loop regions. So these loop regions have been previously heavily implicated in antibody bindings. The N1, the N3, and the N5 loop together form the antigenic supersite, which has been defined in some previous work. So for the last few minutes or so, I'd just like to talk about a particularly interesting persistent infection. And this was a BA2 persistent infection. It lasted 213 days. And what was really interesting about this one is that we saw the emergence of multiple deletions within the N-terminal domain, all of them within these loop structures, specifically N1, N3, and N5. So we saw the emergence of del 257 to 264 quite quickly during the persistent infections. To each of these lines in the alignment represents a subsequent sample in the persistent infection. We also saw the emergence of N1 and N3 loop deletions. What's really interesting about these is that the deletions seem to expand. So we first see emergence of a deletion 14 to 20 within the spike protein. We then see the emergence of this longer 12 to 20 deletion following that. We see the same dynamic with the N3 loop deletions. We see the emergence of del 144, followed by del 141 to 144, followed by this even longer del 136 to 144. So looking further into this N3 loop deletion, this is all at consensus level. Looking at the subconsensus level, it's actually really cool. So in the first sample, we don't see any reads showing the deletion. By the second sample, we see the emergence of del 144, although it's not at full dominance. There's still some reads which show no deletion. By the third sample, though, we see the emergence of multiple deletions existing within the sample. So we have reads which show del 144, del 141 to 144, this 8 amino acid deletion, del 138 to 145, and this 9 amino acid deletion, del 136 to 144. We then see that del 136 to 144 seems to kind of win out against the other ones. It becomes the dominant variant. So we hypothesize that the spike N-terminal domain during this persistent infection is evolving due to antibody escape. So our next step is to build some proper structures using AlphaFold to fully understand actually what's properly going on here. So in summary, we do see de novo emergence of deletions during persistent infection. Some of these deletions, such as del144, reoccur in many persistent infections, but other ones, such as our NSP1 deletions, don't really seem to be that common at consensus level. And what we hope to explore here using this is different levels of evolution, so the conflict between within-host evolution and what's selected for at the between-host. So the deletions that we observe seem to occur in regions previously linked to immune interactions. And kind of my take-home message for this is that only examining consensus level deletions means that we lose resolution and we miss some interesting and important within-host dynamics. So yeah, I'd like to thank everybody on this slide, as well as the UK-ICN for the opportunity to present.
Thank you, Isobel. Any questions?
Just out of curiosity, are these persistent infections between different people? And kind of like to speculate, kind of chicken and egg question, do you think that these mutations arise and then that is what kind of facilitates a persistent infection? Or do you think that the persistent infection is kind of what gives the virus the time? Because a lot of the sequencing is done, it looked like kind of there was quite a gap at the start, kind of 30 days. So it'd be really interesting to have data that was, it was probably quite rare, doesn't exist, but data that very regularly sequenced at the start to see when these mutations within those 30 days occur.
Yes, the next step is to look at the whole of the ONS COVID infection survey to look at within host deletions. But we also have, so the structure of the ONS COVID infection survey was that they sampled weekly for the first month and then monthly thereafter. So that we do have samples from infections weeks apart rather than months apart. They're defined as acute rather than persistent. But one of my next steps is then to look at those to see if we see evolution, because we have kind of been working under the assumption that that the deletions are emerging during the persistent infection due to antibody pressure rather than the deletions driving the persistent infection. If that makes sense. That's something we need to explore.
Thank you, Dallin.
Very nice talk. I was wondering, obviously the deletions are happening to the RNA and not to the protein. It was nice to see that you've gone to low consensus level sequence to look at minor variant populations. I'm just wondering, can you detect anything that's of a different length in terms of like 1 or 2 nucleotides being deleted before the triplet is gone? So can you see those intermediates, or is it always losing 3 nucleotides?
So we do see 1 and 2 nucleotide deletions, but we've been concerned that they're artifactual because they often seem to occur in homopolymeric regions. When it comes to these deletions, what we tend to see with the persistent infections is that you have one sample where the deletion is at 0% frequency, and then it's at 100% frequency. So you don't really see this intermediate or like 1 or 2 base pair deletions before a triplet, if that makes sense.
I guess that's because it's immediately going to fixation, right? But you know, if you sequence even deeper, would you be able to pick out the mechanism of the deletion? Like, presumably the virus doesn't have a way to specifically delete 3 nucleotides when it's acquiring deletion.
I mean, yeah, so I don't think we could do that with our current data set. I don't think that the sequencing is deep enough. It's something that would be really interesting to explore, but I think you would need to be sampling a lot more during the infection to try and piece that together.
In the interest of time, we're going to move on to the next speaker, but the people who are online have asked if people asking questions could speak a little clearer into the microphone because they're not hearing them very clearly online. Thank you so much, Isobel. Thank you.
So we're going to